All right, engineers, in this video we're going to talk about electrophysiology. So if you guys are here for part 2, I really appreciate it. We're going to go into a little bit more detail and we're going to go over what's called extrinsic innervation of the heart. So we're going to talk about, we already talked about in the cardiac conduction system, like the intrinsic part of it, we talked about how we set a normal sinus rhythm. But now what we're going to discuss is in certain situations, how can we increase the heart rate greater than the sinus rhythm, and how can we decrease the heart rate below the sinus rhythm. So that's what we're going to talk about. If you guys have already watched our videos on blood pressure regulation, we talk about how the nerves are coming out of the spinal cord or from the actual brainstem. So we're not going to go over that pathway. We're just going to look at exactly how the sympathetic nervous system and the parasympathetic nervous system affect the heart rate, and then how the sympathetic nervous system affects the contractility of the heart. Okay, so we're going to look at some of the actual mechanisms of this. So let's go ahead and get started. If we look here, I'm going to have a special receptor there. This receptor is called a Beta1adrenergic receptor. Very, very specific receptor, located within the heart. You can also find it on the JG cells of the kidney too. And what happens is, let's say here, I have a neuron here. This is a sympathetic nerve, right? Let's say that this is a sympathetic nerve. And what happens is this sympathetic nerve is releasing chemicals like norepinephrine and epinephrine. And what happens is norepinephrine and epinephrine are going to come over here and they're going to bind onto this Beta1adrenergic receptor and stimulate this receptor. When this happens, it activates intracellular processes. Like how? So what it does is, it first comes into the cell and activates a G stimulatory protein. Okay? When it activates this G stimulatory protein, what happens is this G stimulatory protein gets rid of GDP and binds GTP, which turns this stimulatory protein on. This stimulatory protein then goes and activates an effector enzyme located on the cell membrane. This effector enzyme is called Adenylate cyclase, right? And what is so special about Adenylate cyclase? If you remember, we said Adenylate cyclase is responsible for converting ATP into cyclic AMP. Why is this important? Because cyclic AMP can go and activate a special enzyme called protein kinase A. Why is this important? Because if you remember, we talked about this a little bit in the blood pressure regulation video, but protein kinase A can target special protein channels. You see these green channels up there? Those L-type calcium channels? Let's say I put a small one right here on the membrane. So let's say I put these L-type calcium channels right here on the cell membrane, right here. So here's an L-type calcium channel, and here's another L-type calcium channel. What happens is this protein kinase A is going to come over here, and it's going to phosphorylate these channels. So it's going to put a phosphate on this channel and put a phosphate on this channel. When you put phosphates on these channels, it's going to stimulate these channels to open. When these channels open, guess who starts flooding in? Calcium. When the calcium starts flooding into the cell in greater amounts than normal, so you already are going to have this calcium coming in normally because of the intrinsic ability of the cell. But then the sympathetic nervous system is going to be like, hey, I'm going to help you out, and I'm going to increase even more calcium coming into the cell. So now you have more calcium into the cell. Now the cell is going to be depolarized more frequently. So what is the whole purpose of this? If we bring more calcium into the cell, this cell is going to depolarize quicker. So because it's going to depolarize quicker, it's going to generate action potentials quicker. Because it generates action potentials a lot quicker, we're going to denote action potentials as APs, this is going to increase the heart rate. What is it called whenever you increase the heart rate very significantly, at least to a point where the heart rate is greater than approximately about 100 beats per minute? This is referred to as tachycardia. Okay, tachycardia. And tachycardia is, again, the point in which there is an increased heart rate, at least when it's greater than 100 beats per minute. So if the sympathetic nervous system is activated, it can increase the heart rate enough to bring the actual heart rate greater than 100 beats per minute. How? Again, phosphorylating these L-type calcium channels to increase more calcium entry into the cell, more than normal, to increase the depolarization quicker, cause more frequent action potentials, which increases the heart rate, which causes this tachycardic effect, okay, which can be normal, sinus tachycardia. It can happen when you're exercising, okay? But now, let's say that you want to slow your heart rate down. Okay, you want to slow your heart rate down. You're trying to relax. You're trying to just chill out, watch some cartoons, okay? What's going to happen? Your parasympathetic nervous system is going to come on. It's going to slow down your heart because we don't need to exert it. We don't want to overexert it. We just want to relax. So acetylcholine is going to be released by these parasympathetic neurons, right? Remember, that are coming from the vagus nerve. And when these parasympathetic neurons, they release a chemical called acetylcholine. Acetylcholine binds onto these receptors present on the membrane. What is this receptor here called? This receptor, we said, was called a M2 receptor, which stands for muscarinic type 2 receptor. When acetylcholine binds onto this receptor, it activates what's called a G inhibitory protein. But remember we said that G inhibitory protein is kind of actually broken into 3 components: alpha inhibitory, beta inhibitory, and another one which is called gamma inhibitory. Whenever acetylcholine binds onto this muscarinic receptor, it activates the alpha inhibitory to separate from the beta and gamma inhibitory. The beta and gamma inhibitory come to the cell membrane and bind onto special channels in the membrane. These channels are special and specific and sensitive to the movement of potassium. What happens is these actual beta and gamma subunit come over here and bind onto these channels. When it comes over here and binds onto these channels, it causes these channels to open up. And guess who starts flowing out? Potassium. As potassium starts flowing out of the cell, what starts happening to the inside of the cell? The inside of the cell is losing positive ions. So positive ions are leaking out of the cell. As positive ions are leaking inside of the cell is becoming increasingly more negative. As the cell starts becoming increasingly more negative, that decreases the rate at which the depolarization will occur. It decreases the action potentials and decreases the heart rate. This effect is it's going to try to hyperpolarize the cell, whereas the sympathetic is trying to depolarize the cell. And this hyperpolarization is going to decrease the action potential rate, which is going to decrease the heart rate. And if the heart rate decreases significantly to the point where it actually goes below sinus rhythm, so less than 60 beats per minute, this is going to be what's called bradycardia. So whenever the heart rate is less than 60 beats per minute, this is called bradycardia, okay? This is called bradycardia. Okay. So we know bradycardia and we know tachycardia. Bradycardia is whenever the heart rate is actually less than 60 beats per minute. Tachycardia is whenever the heart rate is actually going to be greater than 100 beats per minute. What's the next thing? You see this alpha inhibitory unit? There's a specific reason for him. What he does is he comes over here to this adenylate cyclase. He comes over to the adenylate cyclase and inhibits the adenylate cyclase. If you inhibit the adenylate cyclase, you inhibit the conversion of ATP into cyclic AMP. What starts happening to the cyclic AMP levels? They start dropping. As that starts dropping, protein kinase A levels start dropping. As that starts dropping, it starts decreasing the phosphorylation. What is that going to do? That's going to decrease the calcium entry, which is going to decrease the action potential frequency, which is going to decrease the heart rate. That's another way that the parasympathetic nervous system can do this. So 2 things. One is the beta and gamma inhibitory subunit bind onto potassium, voltage-gated potassium channels, which are then going to open and potassium can flood out of the cell, losing positive ions, making the cell more negative, hyperpolarizing the cell. And if the cell hyperpolarizes, it decreases the rate at which it actually can generate action potentials, which decreases the heart rate. And if it goes less than 60 beats per minute, it's called bradycardia. Another way is it can activate this alpha inhibitory, which can bind onto adenylate cyclase and inhibit the conversion of ATP into cyclic AMP, which decreases the protein kinase A levels, decreases the phosphorylation, decreases the calcium entry, decreases the frequency of action potentials, which decreases the heart rate as a result. Okay? That's the parasympathetic effect. Now, who is supplying the actual nodal cells with the parasympathetic? You have to remember this is the vagus. Okay? You have to remember that this is the vagus nerve. Okay, and then the actual sympathetic nerves are coming from the chain ganglia or from the superior, middle, and inferior cervical ganglion. Okay, so they're coming from T1 to T5, from the Chan ganglia, superior, middle, inferior cervical ganglion, the sympathetic fibers. And the epinephrine is also released from the adrenal medulla. Sweet deal. So we know how that's going to affect the heart rate. How does the sympathetic nervous system, one more thing. What is it called whenever you increase the heart rate? So the sympathetic nervous system is trying to increase the heart rate. That is called, whenever you try to increase the heart rate, it's called positive chronotropic action, okay? But then the vagus nerve, via the parasympathetic nervous system, is trying to decrease the heart rate. If you try to decrease the heart rate, what's that called? It's called negative chronotropic action. We'll talk about this in more detail whenever we get to cardiac output. But again, just giving you that relationship there. Now, the sympathetic nervous system also has receptors on the contractile cells, not just on the nodal cells. It's a beautiful thing. So now what can happen? Same thing. Let's pretend that actually guy over here, that nerve, same thing, he's coming over here, and he's releasing what chemicals? He's releasing norepinephrine, and let's say that there's also the release of epinephrine from the adrenal medulla, right? So there's also epi here. These guys are going to bind onto this, what receptor? It's still the same receptor, the beta-1-adrenergic receptors. So these are your beta-1-adrenergic receptors. When these guys bind, they stimulate this G protein coupled receptor, which does the same function here. So it's the same action. What's the overall effect here? What happens? Let's actually show you. Real quickly, fastly, right? G stimulatory protein here does what? Activates, what's this enzyme here? Adenylate cyclase. Adenylate cyclase does what? Converts ATP into cyclic AMP. Cyclic AMP activates protein kinase A. If protein kinase A levels increase, this is where it becomes very critical. So as the protein kinase levels increase, two things happen. Let's make this pink so that we can see how important it is. Look at this. The protein kinase A comes over here, and he does two things. One is he phosphorylates these channels here on the sarcoplasmic reticulum, very special channels that consist of a special protein. So one thing is he phosphorylates these channels, which consist of a special protein. What is this protein here called that's kind of controlling these channels? The protein here is called phospholamban. Okay, that's a heck of a name there. Sounds like, something off of the Flintstones, but like phospholamban. Okay? So there's this protein called phospholamban, and what happens is when you phosphorylate the proteins, these phospholamban proteins, it stimulates these channels right here. You know what this does? It opens up the channels on this membrane and sucks in a lot of calcium ions. Sucks a lot of calcium ions into the sarcoplasmic reticulum. What is the purpose of this? If we bring a lot of calcium ions in here, we're trying to increase our organellular storage of this calcium. Why is that important? We're going to see in just a second. So one thing is the protein kinase A phosphorylates the phospholamban proteins on the sarcoplasmic reticulum, sucks more calcium into the SR. That has a significant purpose. The second thing that happens is the protein kinase A comes over here. You see these channels here? These L-type calcium channels? You know how important they are? Again, they come over here and put phosphates on these voltage-gated calcium channels. What happens? More calcium than normal is coming into the cell. So more calcium is coming into the cell. If more calcium is coming into the cell, we have a lot more calcium rushing in. As a lot of calcium rushes in, it stimulates, again, what are these receptors here called? The ryanodine receptors type 2. Guess what though? We have so much calcium in this sarcoplasmic reticulum now. Why? Because the protein kinase A phosphorylated the phospholamban to suck a lot of calcium in there. So now, also what happened? Protein kinase A phosphorylated these calcium channels, the L-type, to bring more calcium in. As we bring more calcium in, it stimulates the ryanodine receptor type 2, and now there's so much calcium in the SR that we release out even more calcium than normal into the sarcoplasm. More calcium means more interactions with the troponin, which moves the tropomyosin. If you move the tropomyosin, you're going to have more actin-myosin interactions. What is that called? You're going to have significantly increased cross-bridge formations. If you have more cross-bridge formations, you have more power strokes, more sliding of the myofilaments. So that's going to increase the actual contractions. If it increases the speed of the contractions, increases the rate of the cross-bridge cycling, it's going to increase the actual pumping action of the heart. And what is this going to do? We'll talk about it more in cardiac output, because you know that whenever you increase the pumping action of the heart, the contractility of the heart, it increases what's called your stroke volume. And then as a result, it can increase your cardiac output. And this, as a result, can increase your blood pressure. Okay? So that is how the sympathetic nervous system can affect the contractility. Because if it affects contractility, it's going to increase the actual cycling of the cross-bridge formations. It's going to have more contraction speed, more heart pumping activity, increase in the stroke volume, which is the volume of blood pumped out of the ventricle in 1 heartbeat. And then it's going to increase the amount of volume of blood that's being pumped out of the ventricles in 1 minute, and that's going to increase your blood pressure. Okay? So sympathetic nervous system can not only speed up the contractility, but increase your blood pressure. At the same time, it also increases the heart rate. What's the relationship of the heart rate with blood pressure? It's directly proportional. Remember, we said that if you remember here, we said cardiac output is equal to heart rate times stroke volume. If you increase the heart rate, you increase the cardiac output. And then we said that the cardiac output, we actually can rearrange this a little bit better, blood pressure is equal to cardiac output times total peripheral resistance. If I increase the cardiac output, I increase the blood pressure. So it's a beautiful thing. And then same thing with the actual acetylcholine. Acetylcholine is acting on the muscarinic receptors, decreasing the heart rate. If you decrease the heart rate, you decrease the cardiac output. If you decrease the cardiac output, you decrease the blood pressure. So these are the beautiful ways in which your sympathetic nervous system and parasympathetic nervous system can affect the actual nodal and contractile cells. But again, parasympathetic only on the nodal cells. Sympathetic, nodal, and contractile. Okay. Last thing that I want to talk about here is I want you to see graphically how this is represented. So let me show you here for a second. Let's say that I bring here in the graph, I bring in, let's do it like this. I'm going to represent black as normal for the heart rate here. So let's say here's my heart rate. I'm coming up here, coming down, coming up, coming down, right? And then same thing here. Just say this is the normal pacing of the heart rate. Okay, this is what's going to look like on the graph. Depolarization, repolarization, depolarization, repolarization. You get it. But then let's compare that with the sympathetic nervous system, and let's do that in this brown color. Now, the only thing that's going to be different is we're going to have action potentials much sooner. And it's going to repolarize pretty quick. And it's going to have action potential sooner, repolarize quickly. And then it's going to repolarize quickly, and then action potential sooner. What is the whole point here? We're going to have the depolarization occurring much faster, and repolarization is still going to be obeyed. We're still going to obey the refractory period. But you're going to notice there's way more frequent action potentials here. That is the effect of the sympathetic nervous system. Now let's compare that with another example, and let's do this one in green, and let's say that the green is the parasympathetic. This time it's going to be a very slow depolarization. So it's going to take a longer time to depolarize. As it takes a longer time to depolarize, what's happening? The rate at which this heart rate is occurring is a lot slower. So the parasympathetic nervous system is going to decrease the action potentials. That's the whole purpose. Sympathetic nervous system increases the rate of action potentials. Parasympathetic nervous system decreases the rate of action potentials. And that's how this affects the heart rate. Okay, so that covers that. Last thing I want to talk about is this whole refractory period. Because it is important that I briefly mention this real quick, is that this plateau phase that we talked about here in phase 2, this plateau phase, this is important because it's about 250 milliseconds. Once it comes from this, it starts going into from 3 all the way to 4 until there's another action potential. This period here is called the refractory period. So this is called the refractory period. It's actually broken up into 3 different parts. Like you can have the absolute refractory period, the relative refractory period, and then another one called a supernormal refractory period. We're not going to talk about that. I just want you to understand the refractory period is when the heart is resting. And it's almost about 250 milliseconds too. So because of that, the refractory period is the time where the heart is resting. This is crucial. You want the heart to rest. In certain weird situations, as this is going down, phase 3, as it's going down, it's getting ready to go into phase 4. If you stimulate the heart enough with very, very frequent, very powerful stimulus, you can take it out of refraction and trigger another action potential. That's called the relative refractory period. So there is a weird one here called the relative refractory period. And this is a situation in which you provide enough stimulus to the heart, you can take it out of refraction and cause another action potential. But you don't want to. You want to obey the absolute refractory period. It's not as much like a skeletal muscle. Okay? Because if you don't, it can lead to problems like tetany, which can become very, very dangerous. If you have tetanic contractions, it can become very, very dangerous. So again, we want to obey the absolute refractory period. Ninja nerds, I can't thank you enough. If you guys watched part 1, you guys watched part 2, I guarantee you're really going to know this stuff now. And I really hope that it helped. I really hope that you guys did enjoy it. If you guys did, please hit the like button, comment down in the comment section, share this video, and please subscribe. All right, Ninja nerds, as always, until next time.